IB Biomolecular Biology Part 4 has its focus on carbohydrates, structure, and function. The essential idea is compounds of carbon, hydrogen, and oxygen are used to supply and store energy. Here's an outline of all the movies available for studying IB molecular biology. Use this slide to reference the part of the topic that you need to review. This movie is focused here. Let's jump in with an IB syllabus statement you've seen before, but modified to reflect carbohydrates. Outline the role of condensation and hydrolysis in the relationships between monosaccharides, disaccharides, and polysaccharides. Even with some new language embedded into this statement, you should immediately feel comfortable with it. The monomers of carbohydrates are known as monosaccharides, and they're joined together by condensation reactions to form disaccharides and polysaccharide polymers. Condensation reactions are anabolic reactions. Enzymes catalyze all reactions in cells. Enzymes control the metabolism of a cell. Hydrolysis is the breakdown of polysaccharides into monosaccharide monomers. Hydrolysis is a catabolic process. Here are two relevant IB syllabus statements that dig into the detail of what you're expected to know and be able to do. Draw the ring structure of alpha-D-glucose, beta-D-glucose, as well as D-ribose. Identify glucose, ribose, and disaccharides from diagrams showing their structure. Here is a diagram of alpha-D-glucose. Notice the hexagonal shape. This is C6H12O6. The carbons are counted from here: 1, 2, 3, 4, 5, and 6. Carbohydrates provide 4.1 kcals per gram, and it is glucose that is the first substrate in cell respiration. Glucose, galactose, and fructose are all monomers of carbohydrates, monosaccharides. They are isomers, as all three have the same molecular formula of C6H12O6. Here is a slide showing an image of alpha-D-glucose and beta-D-glucose. Notice the subtle difference between these two isomers that have the same molecular formula, C6H12O6, but this subtle difference has huge consequences. But let's leave the consequences of beta-D-glucose structure for later in this movie. Stay tuned. As I mentioned, we'll look at beta-D-glucose later in this movie, but let's take a look at ribose. Ribose on the right and deoxyribose on the left are 5-carbon sugars. Pentose sugars. The difference between ribose and deoxyribose is here. Ribose has an oxygen on the second carbon, while deoxyribose does not. Carbons are counted: 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. Deoxyribose is an important part of the monomer that forms the nucleic acid DNA, and ribose is an important monomer that forms the nucleic acid RNA. Here's another look at ribose, and it's an important part of the monomer that forms the nucleic acid RNA. RNA is involved in protein synthesis, the process of translating the code in DNA to the amino acid sequence in a polypeptide. I've spoken briefly about the role of RNA in previous movies. As well, ribose is a part of the molecule known as ATP, a very important energy molecule in the cell. So let's look at condensation reactions that form disaccharides and polysaccharides. Keep a lookout for disaccharide structure. Two monosaccharides will join with the removal of water to form a disaccharide. Maltose, lactose, and sucrose are all disaccharides. Anticipate what's happening here. The monosaccharides will join with the removal of water in a condensation reaction to form a disaccharide. Glucose and fructose join to form sucrose. Galactose and glucose form lactose, a disaccharide found in mother's milk. Here are two important IB syllabus statements. State that the term sugar includes monosaccharides and disaccharides. State three examples of disaccharides. Glucose and glucose join through condensation synthesis to form maltose. Maltose is the third disaccharide I've put before you in this movie. Monosaccharides, recognized here by the hexagonal structure of glucose, join to form disaccharides. Here is a disaccharide. Lactose is a disaccharide important as milk sugar, nutrition for infants. Sucrose is a disaccharide, a sugar transported within the body of plants, ultimately to be used for energy or stored as starch. Here are two polysaccharides, amylose and amylopectin, both starches. Notice the repeating glucose monomers, the repeating monosaccharides. Notice that amylopectin is branched, while amylose is not. Keep this in mind. To wrap up condensation synthesis and hydrolysis in carbohydrates, we would say that monosaccharides are joined by condensation synthesis to form polysaccharides, and that polysaccharides are broken down by hydrolysis into monosaccharides. Examples of polysaccharides include starch, energy storage in plants, glycogen, energy storage in animals, cellulose, which is structural, cell walls in plants, and chitin, which is also structural, cell wall in fungi, and the exoskeleton of insects and crustaceans. As well, we have the disaccharides, sucrose, lactose, and maltose. So now let's focus our attention on polysaccharides, and here's an IB syllabus statement. State one function of starch in plants, cellulose in plants, and glycogen in animals. Starch includes both amylose, which is unbranched, and amylopectin, which is branched. This diagram is of amylose. Amylose coils into a helix, as you can see, and amylose is an energy storage molecule in plants. Amylopectin is branched. Amylopectin is also an energy storage molecule in plants. While both amylose and amylopectin are both serving as long-term energy storage molecules in plants, lipids are actually more suitable for long-term energy storage than carbohydrates. Keep your eyes open for this distinction as we progress through the unit. Now, amylopectin and glycogen are both branched. Glycogen, like amylopectin and amylose, serves as long-term energy storage, but glycogen is long-term energy storage in animals, not plants. Amylose, amylopectin, and glycogen are all polysaccharides, but they are all polar, thus they attract water. When these polysaccharides are packed into vacuoles, water accompanies them. The added water takes up space and makes storage in a limited space less space efficient. Glycogen, like amylopectin, is branched, and thus it takes on a very complex shape as a macromolecule. The high degree of branching seen in glycogen makes glycogen better for packing into the small spaces of vacuoles. So here's the IB syllabus statement that gets under the surface of energy storage, lipids, as compared to carbohydrates. While both amylose and amylopectin are both serving as long-term energy storage molecules in plants, lipids are more suitable for long-term energy storage than carbohydrates. Lipids are nonpolar molecules, thus they do not attract water into the storage space. Storage of lipids is more space efficient. As well, lipid energy content is twice that of carbohydrates per gram. So as I've mentioned, branched carbohydrates make for good packing into small spaces, the vacuoles of cells. As well, these branch points allow for rapid hydrolysis of the polymer to make glucose available for cell respiration. Now, glycogen and amylopectin are both insoluble in water. This reduces problems related to the uptake of water, osmotic problems. Lipids contain twice as much energy per gram as compared to carbohydrates, and lipids are nonpolar. so water does not accompany them into storage. The storage of lipids is more space efficient. Lipids are more efficient in terms of the energy stored per gram of body mass. Now I've come back to this IB syllabus statement because I've not emphasized beta-D-glucose. Beta-glucose is an important monosaccharide in the formation of cellulose, plant cell wall. Your task is to be able to draw the ring structure of alpha-D-glucose, beta-D-glucose, and ribose. Now cellulose is a polysaccharide composed of beta-glucose molecules. The shape of the macromolecule is linear, a fine shape for its structural function as plant cell wall. So here's the difference between alpha-glucose, which is the monomer of amylose, amylopectin, and glycogen, and beta-glucose, which is the monomer of cellulose. Now, you may wonder why such a small difference would result in different shapes of molecules, different shapes that ultimately produce different functions. Now, before moving to the next slide, where we will look at cellulose, composed of beta-glucose monomers, notice the position of the hydroxyl group on the number 1 carbon of beta-glucose and the position of the hydroxyl group on the number 4 carbon of beta-glucose. Watch carefully. So if two beta-glucose monomers are to be joined in a condensation reaction, and the two monomers are oriented as you see here, the positions of the two hydroxyl groups off the number 1 carbon of this monomer and off the number 4 carbon of this monomer do not lend themselves to easy water removal. As you can see here, the two hydroxyl groups are positioned awkwardly apart for the condensation reaction to occur. Keep watching. But if one of the beta-glucose monomers is rotated 180 degrees, upside down, so to speak, the hydroxyl groups are better positioned to join through condensation reaction synthesis. Again, here's another diagram displaying one beta-glucose molecule in one position, and the second one rotated 180 degrees, so the hydroxyl groups are better positioned to join through a condensation synthesis reaction. So, as monosaccharides of beta-glucose are joined through condensation synthesis, every other molecule is rotated, or turned upside down, if I can use such colloquial language. So here is cellulose again. Take a close look at the beta-glucose monomers. This one in one position, this one upside down relative to it, this one in a position, and this one upside down relative to it. As a result of the positioning of individual monomers, cellulose has a linear shape, and that provides the structural function of plant cell wall. And sheets of cellulose come together with hydrogen bonding to form fibers of cellulose. Plant cell wall provides structural integrity to plant cells, assisting in the upright nature of plants who otherwise have no skeletal structure. Here's a micrograph of cellulose. You can see the fibrous nature of the molecule, and with overlapping fibers, cellulose serves people well in making cotton shirts or providing structural integrity with lumber as a building material. So just as a quick note, the exoskeleton of insects or crustaceans and fungal cell wall are composed of a carbohydrate known as chitin. Chitin is also composed of beta-glucose and an added nitrogen group. Now, could you draw these monomers of alpha-D-glucose? Could you draw beta-D-glucose? What's happening here in this image? Could you identify a disaccharide if you saw one? Can you name 3 disaccharides and state the monomers of which they're comprised? Could you name the function of one disaccharide? And what's the function of glycogen and cellulose and amylose? And what's the difference between amylose and amylopectin? And that brings us to the last IB syllabus statement. Use molecular visualization software to compare cellulose, starch, and glycogen. Molecular visualization software is a medium that can be used to communicate structural information about molecules that can be used to understand function. Molecular visualization software supports visual learning through understanding the three-dimensional quality of molecules. In this slide, we can see two images of cellulose. You can see linear chains of thousands of glucose units linked together, and significant hydrogen bonding between the adjacent chains allows the chains to pack into fibers. Cellulose is a structural molecule supporting plant cells as the material of which cell walls are composed. Cellulose exhibits little interaction with water or any other solvent. Cotton and wood, for example, are completely insoluble in water and have considerable mechanical strength. In cellulose, the beta-1,4 covalent bonds result in the linear nature of cellulose. Unlike amylose, starch, cellulose does not form a helical structure. Cellulose's linear structure has high mechanical strength and serves the structural role in plant cell walls. Amylose, starch, serves as an energy storage molecule in plants. You can see chains of thousands of glucose units linked together by alpha-1,4 covalent bonds. Molecular visualization software allows one to rotate the molecule to examine it closely. Starch can form helical structures, which allows it to pack into storage spaces within cells. Energy storage molecules need to be able to store well within plant cells. Glycogen is also composed of chains of thousands of glucose units, but glycogen is branched. Glycogen is an energy storage molecule in animals, and animal cells have even less capacity for storage than plant cells. The branched nature of glycogen allows it to pack tightly within animal cell vacuoles. And that brings us to the end of IB Bio, Molecular Biology, Part 4, on to lipids.